Among the more persistent astronaut anecdotes is one that has nothing to do with breathtaking views or the disorienting thrill of floating upside down. The smell that greets crews returning from spacewalks. They'll open the airlock, stow their gear, and suddenly there it is, a hot, metallic, slightly smoky aroma that some liken to seared steak, others to welding fumes or burnt metal. Nobody's calling it pleasant, but it's distinctive, and it's been showing up in crew reports for years. It's not drifting around in the vacuum outside. You only notice it once the gear is back inside, where air can carry the odor to your nose. The reason comes down to chemistry, not space grilling. In low Earth orbit, the outer layers of the station and any exposed equipment are constantly hit by atomic oxygen, a single atom form of oxygen created when ultraviolet sunlight splits O2 molecules in the upper atmosphere. On the microscopic level, this is a very aggressive substance. It reacts with polymers, coatings, and even some metals, breaking chemical bonds and producing tiny amounts of volatile byproducts. At the same time, drifting through space are polycyclic aromatic hydrocarbons, complex organic molecules found in dust clouds, comet trails, and the remnants of dying stars. When these molecules collide with a suit or tool surface, some stick. A spacewalk puts both into direct contact with the fabrics and finishes of a suit. Back inside, where air can carry them, those reaction products and stuck-on organics create the metallic steak signature astronauts keep talking about. It's easy enough to imagine these molecules as a kind of invisible residue, but the smell provides something solid to work with. If certain odors reliably follow certain kinds of exposure, they can act as informal indicators for engineers studying material performance over time. Detecting new or stronger smells could hint at a higher rate of surface reaction, which might link to faster degradation of components. That's where the anecdote intersects with practical science. The smell isn't just a curiosity, it's physical evidence that space weathering is chipping away at suit layers and station hardware. Atomic oxygen is a known hazard for long-duration missions in low orbit, and tracking how it affects different fabrics and coatings helps guide design choices for future missions. In that way, the burnt steak report doubles as a crude sensor, one that's attached to a living observer instead of a circuit. Astronauts' accounts also underline how environmental data doesn't always arrive through an instrument readout. A shift in smell, a change in surface texture, or a different vibration underfoot can give early clues about changes in conditions. Training crews to notice and report that kind of information is part of keeping people and equipment in working order, especially in places where replacement parts take months or years to arrive. On the ground, scientists can and do simulate the effect. Expose pieces of suit fabric or coating material to controlled bursts of atomic oxygen and selected organic compounds, then check for surface changes and released volatiles. Matching the scent to that produced in space can confirm the chemistry at work and allow teams to predict long-term wear. It might sound niche, but in the design process for something like a Mars-rated EVA suit, it helps determine whether a material stays intact after years of exposure or starts breaking down well before its service life ends. Standing inside an airlock after a spacewalk, the link between that odor and the environment outside is immediate. It's a direct reminder that space is not inert emptiness. It's a place where high-energy particles and harsh radiation are constantly interacting with matter at the atomic level. Each smell is the product of those interactions, embedded into the fibers of a glove or the seams of a suit. And the same physical processes that produce the scent are also slowly changing the properties of the gear itself. The consistency of crew reports on this point, decade after decade, makes it one of those sensory details you almost expect in debriefings. The descriptions vary around the edges depending on mission, materials, and crew, but the broad experience is stable enough that it's practically baked into EVA culture. For engineers, that makes it a reliable if informal data point. For astronauts, it's a small but tangible signal of having been outside. This space odor phenomenon also hints at why sensory science matters in extreme environments. Taste, smell, touch, these aren't just there for comfort. They're parts of a broader feedback system between humans and their surroundings. In orbit, that can mean recognizing the hint of an electrical fire before alarms trip, or noticing a change in vibration that indicates mechanical wear. On a planetary surface, it could mean smelling a chemical in the air that a sensor hasn't yet flagged. That's part of the reason the burnt steak anecdote endures. It's approachable. You might never handle a micrometeorite shield or calculate orbital decay, but you know what a smell is, and you can relate to the idea of a fabric keeping an odor from somewhere alien. It bridges the gap between the abstract physics of space and the human scale of lived experience. And once you've heard it, it's hard not to think about what else is landing on those suit surfaces besides pause and oxygen atoms. In practical terms, the chemistry behind the smell is one small piece of the broader puzzle of space exposure. Every mission that returns usable data on how materials fare under these conditions helps refine the parts, tools, and protective layers for the next trip. The human nose report just happens to be the most direct and memorable version of that data stream. It's a reminder that even in such an engineered, sensor-rich environment, there's room for input from more traditional senses. The other thing to consider is what this kind of residue might mean for biology. Some of the organic chemicals hitching a ride on suits are the same kinds found in meteorites and dust clouds, substances that elsewhere play roles in prebiotic chemistry. Nothing about a spacesuit pod deposit means life is involved, but it does mean those compounds are part of the environment we're moving through. That's relevant if you're thinking about cross-contamination risks between different environments, or about what sorts of molecules tend to survive long space journeys. In the end, the Space Smells Like Steak reports on a breakthrough discovery from 2025. They're a long-running, consistent part of crew experience, backed by plausible, well-understood chemistry. The value is in what they reveal about the interplay between hardware and environment, and in how they signal changes you'd otherwise need a lab to detect. It's one more way the frontier announces itself, quietly, with a signal you process through your own biology. And in that sense, it's a reminder that our planet is not so different. Materials here also tell stories through the traces they carry and the signals they give off. Sometimes those signals are so subtle that nobody notices them for decades, even though they're happening all the time, which makes it all the more surprising when new measurements suddenly reveal that Earth itself has been giving off a constant, invisible product from deep below the surface. Earth's core is leaking hydrogen. Some researchers think the planet might have another kind of hidden output we've only recently learned how to look for. Molecules escaping from far deeper in the Earth than the diagrams in school ever mentioned. One idea attracting attention in geophysics circles is that hydrogen gas could be slowly making its way upward from zones near the core, mantle boundary, released by specific chemical reactions under extreme conditions. This isn't a confirmed, finished picture. It's a working hypothesis built on years of lab experiments, computer models, and a growing stack of deep gas measurements from different drilling projects. The story is less. We've proved it's happening everywhere right now, and more. The conditions to make it happen clearly exist, and we're trying to measure it in the wild. Most people's introduction to gases from inside the Earth comes via a cross-section of a volcano, with arrows pointing to carbon dioxide, sulfur dioxide, and the occasional trace of water vapor. Hydrogen barely rates a mention outside of industrial chemistry, but in specialized studies of the deep Earth, hydrogen shows up as something that could, under the right pressure and temperature, be generated in quantity far below the surface. The core, mantle boundary, is a likely zone for this because it's where iron-rich core material meets silicate mantle rock. Bring any water-bearing minerals into that environment, and the combination of heat, pressure, and reactive metals can split the water's hydrogen from its oxygen. The concept is straightforward enough in chemistry terms. H2O encountering high-temperature iron can lose oxygen atoms to the metal or to other minerals, freeing hydrogen atoms. Those atoms readily pair up as H2 molecules, which then diffuse through surrounding rock. Hydrogen is small, light, and mobile, so given open pathways, like fractures or porous minerals, it can slowly drift upward toward shallower layers. In some scenarios, reactions at depth could have been producing hydrogen for millions of years, quietly adding it to the planet's inventory of natural gases. The difficulty is catching it in transit. Hydrogen doesn't stick around in one place underground for long. It's not prone to forming big reservoirs like methane or CO2, and because it reacts relatively slowly, it can slip past sampling points without obvious signs. Until recently, most detection setups weren't sensitive enough or weren't deployed deep enough to pick it up before it dispersed or reacted with something else. Advances in down-hole probes, gas-tight fluid samplers, and on-site analytical instruments now make those detections more plausible. Even then, each potential find has to be checked to make sure it's not from shallower processes. That's why stories of deep hydrogen usually come framed with a caveat. You might hear about readings from deep faults in old continental crust, or about unusual gas chemistry in fluids from large boreholes, but interpreting those signals means carefully ruling out other sources. It's easy to get a small burst of hydrogen from weathering reactions in rocks just a few hundred meters down, proving that any given sample started out thousands of kilometers below your feet is another level entirely. Part of what keeps scientists interested is how such a leak, if it's happening on the scale some models predict, might intersect with biology. Kilometers underground, far from sunlight, specialized microbial communities live by tapping chemical energy in their surroundings. Some of them can use hydrogen directly as a fuel, reacting it with other compounds to release energy for growth. In hydrothermal vent systems at the ocean floor, similar metabolisms form the foundation of complete ecosystems. If something comparable operates in rock pores or fracture networks on land, then deep hydrogen from mantle reactions could be supporting life we rarely study and often don't even know exists. That biological angle gives the idea weight beyond pure geology. It would mean parts of the biosphere are tied, indirectly, to chemistry happening near the planet's core, independent of surface climate or sunlight. A shift in hydrogen supply at depth could, in theory, alter the balance in those underground ecosystems. It's not an easy thing to measure. Most of those environments are inaccessible without costly drills or mines. But the possibility helps drive interest in mapping where hydrogen is entering the crust. Another reason geochemists are paying attention is that hydrogen doesn't just hang around forever once it reaches the surface. On short human timescales, the flux is negligible compared to the atmosphere's main gases. But over millions of years, the lightest element can escape into space, slowly altering the planet's atmospheric makeup. The long-term effect, if one exists, would depend on the rate of release and on how much hydrogen captures into other compounds before leaving the planet entirely. Instruments and techniques developed for deep Earth hydrogen studies are also relevant to planetary science. The same basic chemical physics applies on other rocky worlds. If you take a planet or moon with internal heat and some water locked in minerals, similar reactions could be generating hydrogen there too. That could feed subsurface microbial communities on Mars, Europa, or Enceladus. Or, if no life is present, it could still influence surface chemistry and atmosphere over time. Comparing what we can measure here with data from space missions gives scientists a baseline for recognizing the signs elsewhere. For now, the researchers involved are careful to describe this idea as a work in progress. They're collecting samples where possible, refining isotope ratio techniques to identify deep sources, and running high-pressure simulation experiments to test reaction pathways. Each line of evidence helps narrow down how likely it is that core, mantle hydrogen is a steady planetary feature rather than a scatter of rare geological quirks. Until the dataset grows, it's best treated as an informed question rather than a universal rule. What's certain is that even a cautious version of this story shows how much we're still probing in Earth's interior systems. School atlases and classroom charts tend to present the deep Earth as stable layers with well-known properties. In reality, interactions at their boundaries can be messy and dynamic, with outputs we're only beginning to detect. Hydrogen from the depths, if confirmed as a sustained process, would be another variable in how matter moves through the planet's interior, links to its surface environment, and possibly to the life it supports. And that's the thread tying it back to bigger scientific surprises. Parts of what we think of as fixed or fundamental often turn out to be linked to processes with very different timescales and pathways than we imagined. Sometimes those links are physical, like gases moving through solid rock. Other times, the hidden connection is written into a system's structure itself, preserved for longer than anyone thought possible. The next story shifts from deep underground chemistry to something far more personal. Material inside every one of us that arrived from a completely different source than the rest of our biological makeup. You're part alien, literally. It turns out a significant part of what makes you human didn't come solely from your human ancestors. A measurable fraction of your genome carries the imprint of ancient viruses, genetic sequences that became part of our DNA long before our species existed. We're not talking about infections you personally fought off. but molecular signatures left by viruses that inserted their genetic code into our ancestors' cells, sometimes so far back in time that mammals hadn't yet evolved their modern form. Standard classroom biology focuses on the human story as a straight evolutionary branch from earlier primates, with maybe a side mention of Neanderthals if the lesson plan allows it. Viruses, when they come up, tend to be framed only as parasites, invaders to repel, not contributors to who we are. What's missing from that picture is the fact that some viruses, particularly retroviruses, have a unique toolset. They can reverse engineer their own RNA into DNA, and with the help of enzymes, stitch that DNA directly into the host genome. If that genome belongs to a reproductive cell, an egg or sperm, those viral instructions become part of the genetic material passed to offspring. Once embedded, they can persist for millions of years, copied along with the rest of our hereditary code in every new generation. Modern retroviruses like HIV still use this capability. The ancient relatives of these viruses did much the same to the early ancestors of primates, and the integrations that didn't harm their hosts could remain indefinitely. Over evolutionary time, these embedded viral fragments have become what geneticists call endogenous retroviruses, or ERVs. They're not independent organisms anymore, just genetic sequences preserved inside cells, often degraded by mutation, but still recognizable as viral in origin. One reason scientists pay close attention to ERVs is that some have been co-opted for essential functions. The clearest example is in the development of the placenta. Certain ERV-derived genes in humans help fuse cells in the placenta into a continuous protective layer between the fetus and the mother's blood supply. Without that cellular fusion, nutrient transfer and immune protection during pregnancy would not work as they do in mammals today. In other words, a vital part of your earliest development relied on a gene that originally came from a virus infecting one of your distant ancestors. These integrations date back staggeringly far. Some ERV sequences we carry today predate the origin of mammals themselves. If you rewind to when our ancestors looked nothing like modern humans, when they were small, fur-covered creatures living alongside dinosaurs, some of those same viral fragments were already writing in their genomes. Bit by bit, they accumulated from multiple infection events in different eras. From the point of view of your DNA, they are simply segments of code to be duplicated as the genome is copied. From our point of view, they're visible proof that human DNA is a patchwork. They are not silent relics either. Some appear to give their hosts an advantage by blocking similar viruses from using the same genetic entry points, like jamming a lock so another would-be intruder can't open it. Others may interact less beneficially, with possible links to certain cancers or immune system conditions if they become abnormally active. Because these sequences are tightly integrated into our genomes, removing them would be neither simple nor necessarily safe. They can be tangled up with genes that we rely on. This dual role as both potential helper and hazard makes ERVs more than just an interesting footnote. Understanding which ancient viral sequences protect us and which might contribute to disease could influence how we design treatments. The same viral mechanisms that once inserted themselves into our DNA are now copied in some modern gene therapy techniques, where doctors deliberately insert beneficial DNA into a patient's cells to address genetic disorders. In that sense, human biotechnology is borrowing from viral biology, turning an ancient process to a new purpose. Recognizing that our genomes are mosaics has broader implications for how we understand life's history. Our double helix includes not just the legacy of our own species and our primate relatives, but also material traceable to far older life forms, ancient organisms, bacteria, and these viral fragments. The genetic code you carry is not a static instruction manual authored by one lineage. It's a document shaped by repeated outside contributions over deep time. And unlike the rapid change seen in microbes, which can adapt in days or weeks, most of these viral integrations are fixed on evolutionary timescales. Once an ERV becomes part of the germline and spreads through a population, it can remain recognizable for tens of millions of years. That's the opposite of the fast evolving viruses and bacteria we watch in real time today. Your genome therefore holds two overlapping histories: slow, inherited sequences of ancient origin and the constantly shifting microbial world that interacts with you from the outside. Much of what's known about these elements has come into focus only recently, as genome sequencing technology has become faster, cheaper, and more accurate. By scanning enormous stretches of DNA for patterns and motifs, researchers can pick out viral sequences and trace their origins. The latest advances have allowed teams to map these integrations in far greater detail, confirming which functions they've been absorbed into and how far back they go in the tree of life. In some cases, a viral sequence has been so altered by mutations that it's inert. In others, enough remains intact to be expressed as part of a working gene. Presenting this to a general audience works best without overstating the case. It doesn't mean you're part alien in the sense of being fundamentally non-human, and it doesn't mean an ancient infection is waiting to re-emerge fully intact. What it does mean is that the boundary between self and other is blurrier in evolutionary terms than it might seem. The line between an invader and a collaborator has shifted repeatedly over hundreds of millions of years.